On TRPV6 as a genetic factor in African American prostate cancer disparities. I would like to acknowledge our advisor, Professor Patricia Francis Leon, and Senior Data Scientist at Microsoft, Dr. Bob Horton, for all their help with our research study. Next slide, please. In this presentation, we'll be reviewing the background and theory for our study, pre-processing techniques, model building, top SNP results, PCA with varimax rotation, and then we'll share our conclusions and expand on our future plans. Next slide, please. So cancer disparities. It's well known that a significant amount of the African American diaspora originated from the sub-Saharan region of Africa. It's also well established that African Americans have a higher incidence of cancer in comparison to other ethnicities. Moreover, we see here in Ward et al.'s publication that the incidence of prostate cancer mortality is markedly higher between African Americans and European Americans. Although there are environmental, socioeconomic, and access to healthcare aspects to consider, there is also a lack of genomic evidence to help understand or ameliorate the problem at hand. Next slide, please. So, background on lack of genomic information between groups. So a majority of genomic studies use SNPs to determine differences between groups. Furthermore, a SNP is a single nucleotide polymorphism, which constitutes a single point change in the DNA. SNPs constitute a lot of variation we've seen between individuals and groups. From the paper, clinical use of current polygenic risk scores may exacerbate health disparities. We see that the red in this graph denotes a majority of participants in genomic studies being European, whereas a fraction of the participants in purple are African. Moreover, to the figure to the right, we can see that the prediction of phenotypic traits through biobanks will result in almost a perfect accuracy for Europeans, but the least accurate for Africans. So considering that there is a lack of SNP data for African Americans, we can use data supplied by the NIH to distinguish phenotypic differences. To conduct our analysis, we will plan to examine SNPs of African American men with prostate cancer in context to their TRPV6 phenotypes. Next slide, please. Targets. So it's well established that TRPV5 and TRPV6 play an active role in calcium transport across epithelium tissue. Presently, many people have developed the derived mutation for TRPV6 at such a high frequency that the derived has become the reference and the ancestral has become much less common. We hypothesize that African Americans from the sub-Saharan region of Africa may have retained the ancestral allele for TRPV6 due to lack of viable dairy farming in the region. Therefore, without the introduction of higher concentrations of calcium into their diet, the ancestral allele remained predominant in that particular region and potentially deleterious when engaging in a calcium-enriched diet. So, next slide, please. Developments. So when examining unique deviations in SNPs, we use the genome construct GRCH38 as a reference, where the predominant allele is known as a reference allele and the less common is known as the alternate allele. In our study, we focused on probes that elucidate the identity of TRPV6 phenotypes in context to African Americans and European Americans with prostate cancer. With this data in hand, we may gain insight into men who have an at-risk genotype for TRPV6-associated prostate cancer. Moreover, the elucidation of a personal genetic identity among populations can empower medical decisions and best practices. Initial improvements to our pipeline included integrating Illumina file reading capabilities, SNP database expansion, and the identification of ancestral mutation counts. However, we have not yet secured our university permissions to use our NIH patient data. In this case, we performed a series of Gunter and Reich episodic disease models. An episodic disease model is where gene interactions are associated for a given effect. So for these models, we retrieved and processed real SNP open source data. From our 1.3 million SNP database, we use our TRPV5 and TRPV6 SNPs to create a cancer phenotype designation in alignment with our hypothesis of dysregulated calcium ion transport promoting prostate cancers. Our leading results were derived from epistatic, recessive, and multiplicative models, which yielded high-ranking scores for our SNP targets. Now I'll hand it over to Dasha.
Thank you, Patrick. So let me give you an overview of how the pipeline we use for detecting genomic associations works. The pipeline is broken down into three following steps. First is the pre-processing, in which, while pre-processing the genomic data, we build our SNP database from 1.3 million single-point mutations cataloged by dbSNP. We read in genomic data taken from entire human genome of human subjects. Each subject is then labeled as a disease state, in our case, cancer or not cancer. At the end of the pre-processing step, we have mutation count at each step for each user. Second comes the modeling, in which we are using machine learning, where the machine learns from example. We split the data into train and test sets, and then train the machine to distinguish cancer from not cancer using the genomic data in the training set. Finally, in the prediction stage, we then predict cancer status for the subjects in a test set and evaluate how well the machine has learned. Next slide, please. For data pre-processing, previous members of our group investigating genetic traits developed a pipeline that our team added to. The pipeline with genomic data from popular platforms Ancestry.com and 23andMe. We have been granted approval from NIH to use Illumina genomic data. And in preparation of that, as you can see in the picture below, we added a capability to read the SNP and subject data. Next slide, please. For data modeling, in the pre-processing stage, we have 1.3 million total SNPs in our database and 610 total subjects initially. The number of subjects then came down to 435 at the end of pre-processing because of many duplicate files, or the genomic data of subject was not in the format of Ancestry or 23andMe. For modeling, around 800,000 SNPs were removed for each phenotype since those SNPs had too many missing subject observations. 53,000 candidate SNPs were identified by a pipeline out of 1.3 million SNPs. Also, 12 subjects were drawn due to too many missing observations, and hence we had only 423 subjects left. Next slide, please. We used ElasticNet and random forest for selecting the top SNPs from the candidate SNPs. We chose our methods that rank variables by importance so that we could get importance rank for a TRPV6 gene, the one highlighted in yellow, which has RSID ending in 7667. As you can see, we got better results with ElasticNet, where we got a TRPV6 gene in the top 10, as seen in the right image, than the random forest, which got a TRPV6 gene at 369 position, as seen in the left image. We chose the result from ElasticNet as it is known as better at picking out important variables out of a large number of variables that are just noise. I now pass on to Daniel, who will be taking over.
Thank you, Dashan. So for our data modeling, the approach that we make use of was principal component analysis with very match rotation. You might be asking yourself why we make use of this approach and not traditional principal component analysis. The reason behind it is because we want to have more interpretability in order to be able to understand our SNP contribution and for us to understand whether or not a patient will be classified as cancer or a patient with non-cancer, which led us over here to our principal component regression analysis. And from this principal component regression analysis, we wanted to see how well our model was being able to separate these two classes, which will be displaying in our 2 and 3 rotated components plots. So over here, what we're looking at is our PCA with very much rotation using 2 components. Over here, as Dashan previously mentioned, we're looking at our top 10 SNPs from the ElasticNet model, where the one of our interest is the one ending in 667, so we're TRPV6 SNP. From here, we see that there's a 90% contribution in strength on this rotated component number 1. So that led us to use principal component regression, where over here we see that there's a positive relation, and this means that our rotated component number 1 it will provide us with a high probability with patients with cancer. Here, from our rotated component number 2, we see that it has the opposite direction, which in other words, it means that the rotated component number 2 will let us know or classify patients with cancer at a lower probability. For our goodness of fit, we make use of pseudo R-square, which is 61%. You might be wondering why is it a little bit low. The reason behind is because this analysis is done in an agnostic approach without taking into account important factors such as biological or socioeconomic. So then we were able to plot this information in these two rotated components, where the x-axis has our rotated component number 1, our y-axis has rotation component number 2, and these dots represent our patients, where our color code based upon cancer versus non-cancer patients. The red ones are the cancer, the purple ones are the non-cancer patients. From here, what we expect to see is a line of separation of how well our model does. So over here with my cursor, I'm looking at how well our model is splitting these two classes. So it's a little bit hard to visualize this information in a 2-dimension, which allows us to make use of a third rotated component, which looks like this. And through this visualization, we're able to interact with it and have a further more analysis in order to see if our model is able to actually classify, to split these two particular classes. So that's a little bit about how we were able to do our analysis. And now I'll hand it over to Patrick to let you know about our conclusions and future work.
Thank you, Daniel. So our pipeline can select genes and SNPs associated with disease. Moreover, it works with epistatic and main effect disease models. We've shown our process of going from 1.3 million SNPs down to 4,000, and then being able to subset the most influential SNPs. Not only does this pipeline work for prostate cancer, but we also can generalize the pipeline for other disease classifications. We have focused on interpretable methods so as to learn how SNPs are associated with disease. And this summer, we will commence analyzing our NIH genomic data labeled for epithelial cancers. The exciting portion of our NIH genomic data is that the African genome is well represented and balanced in the NIH dataset. Overall, we hope to find genomic associations to inform the treatment of African Americans as it pertains to epithelial cancers. Thank you again for all your time, and now we can open up the floor for any questions.